Hey guys, come with us to Care Welkins. We're here for a lab visit. I'm gonna show you everything there's to know. Let's go. Okay, so we're starting off with the blood grouping section, which is where we're going to determine the patient's blood type based on the presence of certain antigens on their red blood cells. So here the technician was preparing samples by adding antibodies of each blood type. So if there is agglutination, which is like the clumping together, that means that antigen is present. For example, in this clip you're going to see the last three have agglutination.
So microbiology is all about identification of bacteria and sensitivity. So if the doctor is suspecting there's any infection, so they'll send the sample. So we have urine samples, we get tissues, fluids, pus, swabs, bones, anything you can imagine of. They're going to send the sample to us, and then we're going to do the culture. So for culture, we have different media plates that we use. So that's our sterile area. Okay. You can just have a look. That is our sterile area.
Okay.
So for the sterile area, we're going to prepare all our media inside, and this is our working area. We have different type of media to support the growth of different type of bacteria. So for example, this is the clay plate. Okay, I can show you how it goes. So when the bacteria goes, you can have different.
You remember this?
Yeah.
Oh yeah.
I'm
talking about
like this. We have something interesting also. This is strep pneumo.
Oh.
So we culture them on different media plates. Once we do the culture, we put in the incubators. The incubators are set at 37 degrees Celsius because we are dealing with human samples. Okay, so you want to keep it at the optimum temperature. And after the culture is done, we are going to do the plate reading. So whereby we are going to read, we are going to see what is growing. We are going to identify the organisms and then determine whether they are significant or not. So you can use the disc diffusion method whereby these are antibiotic discs. These are antibiotic discs with known concentration. So if the bacteria is sensitive to the antibiotic, you're going to have a zone.
Nice.
You see, it's not growing around the disc.
Yeah.
Means it's working against it. But the zone size is going to determine whether it is sensitive, intermediate, or resistant. So we follow the CLSI guideline.
Okay.
You have different guidelines.
This is our coagulation machine.
Okay.
Full blood count analyzer.
Okay.
Go for same. It's N550. Okay. Here we have our Savior machine for hemoglobin electrophoresis.
Okay.
Two departments merged in a single area. So we have two analyzers. This is our main biochemistry analyzer, and this is our immunochemistry analyzer. So on the biochemistry analyzer, we do tests like glucose, creatinine, renal function, liver function, lipids, cholesterol, calcium, magnesium, in brief. And here on the immunoanalyzer, we do more specialized tests like thyroid, hormonal tests like thyroid. We do also hormonal tests for fertility for both males and females. We do also tumor marker testing, which is for cancer screening.
Hey, me again. So then we went to the histopathology, which is where tissue samples are processed, stained, and then examined under a microscope to diagnose diseases. And then lastly, we went to the phlebotomy section, which is where patient samples are initially collected. And if you're afraid of needles, look away now. Samples include blood, urine, semen, cerebrospinal fluid, etc. And with that, we have concluded our tour of the Care lab. Thank you to Care for welcoming us, and thank you for watching this video. Bye.